Okay, so everyone see the slides, okay? Interrupt me anytime if you want me to slow down or re-explanation to you. So from your course description, from the syllabus, I make some change. Let me summarize the unit 2 for you. So we spent 3 lectures on general feature of bacteria. We learn cell structure, cell function, bacterial pathogenesis, bacterial metabolisms. We learn antibiotic and how bacteria resist antibiotic as an introduction. So hopefully tomorrow, half of the lecture tomorrow, we will finish everything. For pathogen itself, we divide bacteria to be 2 groups. This is my flow to help you remember easily. One is usual bacteria, another one is unusual. We put unusual is in the last lecture. Okay. For usual bacteria, we separate bacteria by gram staining. Okay, gram positive and gram negative, 2 gram only, right? The gram positive, then we observe microscopic level by cocci and bacilli. Everyone know that, right? If we found it's cocci, then arrangement of cells is very useful for characteristic, for classification of bacteria. If we found gram-positive cocci arranged in cluster, then you probably think about Staphylococcus, right? If they arrange in chain, then we think about, oh, so many, some people coming. All right. If the cells arrange in chain, then you probably think about Streptococcus. Well, if bacteria, you observe bacteria by gram stain under microscope, and you see gram-positive bacilli is a rod, then we further classify by spore formation or non-spore formation. Okay, we learn spore formation and non-spore formation in lecture 14, 15. After that, if we found it is gram-negative can be cocci. We learn in lecture 16. This is the new arrangement. And some half of them we learn gram-negative bacilli. Gram-negative bacilli is a huge group. So then you spend like almost 3 lectures to learn about gram-negative bacilli. So there are so many species and they're mostly toxic, okay? Spirochete, actually we never determine them as gram-positive or gram-negative, but because they show up like outer membrane, so we classify them as gram-negative. So there are no gram-positive spirochete, okay? All spirochete are gram-negative. Then, but most of the spirochete that infect human, they are, well, the important one, just like 2 or 3 species, so few. Then we spend the same lecture with anaerobic bacteria. And anaerobic bacteria is another one that's very important. And the final lecture, we talk about unusual bacteria. That's the big picture of this unit, okay? Okay. So let's start from the basic prokaryotic cell. So prokaryotic cell structure, this is so basic. You know it. It's a cell that no prostate nuclear membrane, okay? Since they have no nuclear membrane, the genetic material in the cells, they blend in cytoplasm, but they're not mixed together. They have area. The area that DNA or chromosomes stay, we call the nucleoid. We talk about that in the last part. The way of cell division are not processed, not undergo mitosis, meiosis, but they use binary fission. Binary fission coming from chromosomes split into 2 copies, and some part of the nuclear membrane start concave together and then separate these compartments into 2 small cells, and these small cells kind of swirling bigger and bigger. That's determine the cells growing. Based on prokaryotic cells, all prokaryotic, well, I would say most of prokaryotic cells, just 99%, present cell wall. And cell wall make bacteria has shape, okay? Based on the shape of bacteria, we classify bacteria into 3 different shapes. The one is sphere, look like the cell looks sphere like a ball. We call coccus. Plural is cocci. Sometimes we probably see coccus arranged in so many shapes. 2 coccus together, when they get cell division and then the cell is not split from each other, we call diplococci. Okay, if they arrange in chain, we call this pattern as streptococci. Okay, strep means chain. If they arrange in 4 cells attached together, we call tetrad. Okay, or sometimes the tetrad duplicate again, get like a cubic 8 cell together. We call Sarcina pattern. For example, in the genus Sarcina, if they group in a cluster, a clumpy cluster, like this picture, we call staphylococci. This is the name of pattern. But don't confuse staphylococci also the name of genus. Okay. Another shape is the rod shape. You can see single bacilli, the rod called bacilli, okay? Sometimes you probably see 2 rods attached together at the pole, not attached like this, but attached like at the pole position. We call diplobacilli or streptobacilli. And this one is probably difficult for you in the lab if you observe under microscopic level. Sometimes the cells more or less like cocci and more or less like bacilli because it's super short rod. We call coccobacilli. It's hard to decide coccus or bacillus, but we group in the bacillus because a bit longer than sphere. Spiral can be everything since curvy or spirillum or spirochete. Spirochete is thin. Spirillum is quite fat. The cell's fat diameter of spirillum is quite big. Okay. So in this lecture, we classify the cell structure into 3 groups. Okay. The first group is the cell envelope. Envelope determine the area that say this is cells. Okay, we start from cell envelope and then we go to the external structure. External structure is mean everything that project out of the cell and meet directly to the environment. And internal structures that mean everything that stay inside the cell envelope. Normally we call cytoplasm. Okay. Let's start from the basic one first, plasma membrane, the cell envelope. This is very similar to eukaryotic pattern. So plasma membrane can comprise lipid, phospholipid, okay? and protein. Protein can be attached to lipid or another protein attached to the protein in lipid. Okay, the protein that interact with the lipid and immense itself in the lipid bilayer, we call the membrane protein. Okay, the protein that the membrane protein that project some part of the protein outside, some part of protein inside the cytoplasm. Like this, we call transmembrane protein. Okay. This picture is I steal from a textbook that show eukaryotic membrane. In fact, there are no glycoprotein present on membrane, on the protein like this. Glycoprotein, well, I would say rare bacteria contain that. Glycoprotein like this, they need to add the sugar in the protein in Golgi apparatus, but bacteria don't have that. Okay, so this is the marker of eukaryotic membrane. All right. So main function of membrane is osmosis. So permeability of substance. Let's think about osmosis. Bacteria has cell wall, right? When they immerse in isotonic solution, isotonic is mean like concentration of solution equivalent to concentration of the cell cytoplasm. So cells remain the shape like normal shape. But if we move bacteria to the new solution we call hypotonic. Hypotonic is solution that lower than cytoplasm. So water from outside start osmosis to inside, just move inside the cells. And if some bacteria that cannot tolerate osmotic pressure, cell burst, so they lysis. But most bacteria has cell wall, particularly gram-positive, right? You learn gram-positive, that means bacteria have strong cell wall. They can tolerate the high osmotic pressure. But if you add something more, make the solution hypertonic, they pull the water back to the environment. So plasma membrane and protoplast, that means plasma membrane and cytoplasm will shrink. But if you stain the cell, you still see the shape of the cell remain the same as bacilli, as cocci, whatever, because of cell wall rigidity. Okay. Main function of plasma membrane is permeability. Select some substance, move in and move out. If some substance that cannot move directly by diffusion across membrane, they need some protein, okay? And also these protein not just only channel for pull something in and get something out. These protein can be receptor, okay? Can be receptor, can be a ligand to let bacteria attach to the environment, okay? And also the membrane of bacteria can working for energy conservation, can help bacteria generate a lot of energy. Energy in this case mean ATP, okay? For channel protein, this is important. Some substance can bring some type of channel protein can bring substance pass through the protein without energy. We call facilitated diffusion. Okay, some of them need energy, so that means active transport in bacteria. For example, well, I will give you example when we reach that topic in bacteria. So from membrane, we move a little bit to cell wall. So cell wall is the very rigid part outside of the membrane. In bacteria, as you know, there are two different types of cell wall. One is gram-positive cell wall. If you see this picture, the cartoon, the brown one is membrane. The purple one is cell wall. Cell wall is quite thick, much thicker than membrane, okay? And cell walls exposed to the outside of the cells. Composition of cell wall contain a polymer. You need to remember this. This polymer called peptidoglycan. Unlike the other organisms, like plant use cellulose, fungi use something else, but bacteria use peptidoglycan. Okay, it's main substance in cell wall. Sometimes they probably have protein attached to peptidoglycan. Okay, and one important thing, you see the red rod here. This is teichoic acid. Teichoic acid is a molecule that presents only in gram-positive bacteria. Okay. Actually, some teichoic acid attached to peptidoglycan, but some teichoic acid attached to lipid, phospholipid in the membrane. So we call lipoteichoic acid. Okay. And then we move to gram-negative bacteria. Gram-negative bacteria, if you see this cartoon, you see peptidoglycan, the purple layer is so thin, but they have another layer, the red one, called outer membrane. So bacteria in this group, gram-negative, has double membrane. One is plasma membrane, another one is outer membrane. In between outer membrane and plasma membrane, they show a space we call a polyplasmic space or polyplasm. Okay. In this area, you can find peptidoglycan, a thin layer, maybe one, not more than 5 layers of peptidoglycan, compared to gram-positive that may be more than 40 layers of peptidoglycan. Okay, so the outer membrane of gram-negative contain lipopolysaccharide. Okay, this part, lipopolysaccharide. This is very important. Lipopolysaccharide is the combination of polysaccharide and lipid. Okay, the lipopolysaccharide we call it LPS. Okay, we abbreviate it as LPS. It is one component of gram-negative that function as toxin. Okay, and on well, this picture don't show the molecule. On the outer membrane here, they have a channel molecule, channel protein, we call porine protein. We talk about that later. Porine is very important. Well, when substance move from inside of the cells to polyplasm, they probably use this picture is wrong. Porine is not present here. Porine is present on the outer membrane. Well, I probably need to change this figure next time and teach. Okay, porine is present on outer membrane, not cell membrane. Okay. When they pass channel to polyplasm, the substance will pass porine outside. Okay, so porine is kind of gate to allow the big molecule move in and move out. Apart from gram-positive, gram-negative, we have another type. Or let me see. I summarize things here. You can go to read this is in-depth teichoic acid and lipoteichoic acid. This is characteristic of gram-positive cell wall. Okay. So let's see peptidoglycan. Peptidoglycan is heteropolymer. Heteropolymers mean the polymer of A, B, A, B, A, B linked together like this, okay? One unit is we call NAG, NAG, that stands for N-acetyl glucosamine, okay? And NAM is N-acetyl muramic acid. So we have two types of sugar molecule linked together, NAG, NAM, NAG, NAM, like this. N is mean like there's so long of this, right? At the N-acetyl muramic acid, they link with another 4 amino acid. This amino acid is not just common amino acid in metabolism, but a derivative of the amino acid, okay, for peptidoglycan. So everyone can imagine the structure of peptidoglycan, okay? Well, when peptidoglycan like this polymerize in the long polymer, you probably think about the string with small drop of amino acid. And then one string of polypeptide, polysaccharide, and another polysaccharide that has amino acid. They need to link together to make it more tight, okay? So the link together of this happen between amino acid, okay? We call peptide cross-link. In gram-negative bacteria, in this picture, E. coli gram-negative bacteria, they link together from alanine to another amino acid, third position. But in gram-positive, they add 5-glycine. We call glycine bridge. But this is not the characteristic of gram-post and gram-neg. That is because the peptidoglycan are well studied in E. coli and Staph aureus only. So we know in detail just only 2 species, okay? But we assume the other gram-positive should be like this. Glycine, number of glycine may vary between gram-positive amino acid, but gram-negative have nothing, all right? Apart from that, here, this is gram-negative. Gram-negative has cell membrane, outer membrane, and porins stand on the outer membrane. In between outer membrane and inner membrane, the cell membrane, we call polyplasmic space, okay? Peptidoglycan stay in between of that, okay? And LPS present only in gram-negative. Remember, LPS gram-negative, but teichoic acid gram-positive. Porin found in just only gram-negative, right? See, the LPS here. This is molecule of lipopolysaccharide. They contain 3 parts. The first part is lipid A. This part immense in the membrane of outer membrane. This part is called polysaccharide. Called polysaccharide means several bacteria, they show up with almost similar pattern or maybe slightly different of the polysaccharide pattern. But this part we call old specific polysaccharide is vary from species or strain to strain, okay? And this part is also the antigen of bacteria because they project out of the cells. They located on the outer surface, okay? When these bacteria go inside human body, so immune response will recognize this part because different bacteria show up with different pattern, that thing. But the lipid A itself here, the yellow part, they function as toxin. This toxin called endotoxin. The endotoxin is very, very potent. It is heat stable because they're made of lipid. So heat cannot destroy structure of lipid, okay? When bacteria, gram-negative bacteria that have lipid A go inside body, when immune response destroy the cells, cell lysis. When cell lysis, they reveal the part of lipid A outside of the cell compartment, then human hosts will get effect from toxin. Okay, this is one of the important gram-negative toxin. Then porine protein. Okay, porine is part of gram-negative outer membrane. Here, itself can be a beta barrel, but 3 barrel packed together. So one porine protein contains 3 channels. Substance will pass through the channel of 3. Okay, this is the function of porine. So based on different types of membrane, different types of cell wall, we develop the method to stain bacteria to be gram staining. Okay. So once we stain, so I suppose everyone know the process of gram staining. So we spend crystal violet, few drop of crystal violet, and leave it 1 minute. Okay, wash by water, and then add gram iodine. Gram iodine is modern. Modern is mean the chemical that link the primary stain. Normally, crystal violet is small molecule. When you stain that, crystal violet go inside the cells in both gram positive and gram negative. Crystal violet itself, be careful, crystal violet itself is basic dye. That means the molecule itself show up the positive charge. They can bind everything that is negative charge in the cells. And most of negative charge in the cells are DNA, are peptidoglycan. Peptidoglycan also negative rich, okay, negative charge rich. Once they go to bind that, gram iodine is modern. They cross-link crystal violet to have big molecule. Once the crystal violet bigger, complex, when you wash by alcohol, the acid alcohol, the ability of alcohol cannot soak the dye out. So in gram positive, you still see the purple in this step. But in gram negative, they have a lot of lipid on the membrane, on the cell wall. So they allow crystal violet complex move out. Okay, and then another point is they have very thin cell wall, so crystal violet can move very easy. In this case, you hard to observe gram negative. That's why we need counter stain. Counter stain is just another stain used as a background or used for differentiating. So when you add safranin 1 minute, gram negative cells will show up the red color. Gram positive cell will show up the purple color. That's gram pose and gram negative. Okay. This is a picture, general picture is gram positive, gram negative, like this. Okay. Apart from gram positive and gram negative, they have another type of cell wall called acid-fast bacterial cell wall. The acid-fast bacterial cell wall look like this. Cell membrane has peptidoglycan. One marker that is different from the other bacteria is the purple one, like this, lipoarabinomannan. This is the molecule found on cell wall of acid-fast bacteria. Okay, and they have a layer of albinogalactan. Okay, they have albinogalactan. No, they never process outer membrane, but has porine. So the cell wall is look pretty much like half gram positive, half gram negative, right? And the outermost has waxy molecule. The waxy molecule is it is acid. Actually, it's fatty acid called mycolic acid. Okay, mycolic acid. Mycolic acid never present in the other type of bacteria except acid fast. Okay, and that makes these acid fast bacteria named mycobacteria. Okay. When we stain mycobacteria, so we use carbofucin. Carbofucin is the red color. Normally, the waxy layer of acid fast membrane, acid fast cell wall, is not allow carbofucin move inside. So in the conventional method, we use heat it up. We use to apply heat by boil water and put the slice over the boiled water and let the cells swell a little bit, and then carbofucin can move inside. But now we use the modern togito. Togito is another substance added to wash up some waxy fatty acid, and then let the carbofucin move inside the cell easily. And then you just watch by acid, okay? Only acid fast staining bacteria, acid fast bacteria, they tolerate acid. So once you wash by acid, the other bacteria that stain acid fast, stain carbofucin, you have been washed out, except the acid fast bacteria. But you can't see the other bacteria, right? You need to get counter stain. Like I said, you stain it for use it as a background or differential bacteria, okay, by methylene blue, okay. In this case, when you observe bacteria, the acid fast bacteria, you show up the red color like this. This is a smear of sputum. So you know sputum, right? Of patient who get tuberculosis. This is the bacteria, causative agent of tuberculosis. Here also, why we cannot use gram stain with this bacteria? Because the cell wall of bacteria in this group, I mean Mycobacterium tuberculosis, is so thick, crystal violet cannot move in. Everything cannot move. Safranin cannot move in. Then when you stain, you probably see bacteria like this. This is the white area that is bacteria, and the dye cannot move in. We call goat cell. Okay, the goat cell. That is the thing about cell envelope. So we spend lots of time talk about cell wall. This is very important because cell wall is the organelle that unique only in bacteria. Okay, so then we move to the external structure of prokaryotic cells. First thing, we talk about flagella. Flagella in bacteria, we have 4 different types. Okay, well, not all bacteria has flagella. Okay, but if some bacteria contain a flagella, if they probably have 1 flagella. We call monotrichous flagella. 1 flagella happen in the pole, cell pole, not happen in the middle of the cell, just the polar of the cell. Amphithicus, 2 flagella, different pole has 1 pole, 1 flagella, 1 pole, 1 flagella. Okay. Lophotrichus, more flagella process in 1 pole, like this. And Perithicus, there are tons of flagella happen everywhere on the cells. Okay, this is Perithicus. Right. Bacterial flagella are different from eukaryotic cells flagella because eukaryote flagella comprise tubulin protein arranged in 9+0. But the bacteria, they not contain tubulin. Bacteria never have beta tubulin protein at all. So the protein that form bacterial flagella is flagellin. Okay. In gram positive, they have one disc. The disc is combination of tons of protein that lay on cell membrane and form a tube structure project out of the cell wall. This is the gram positive flagella. But gram negative flagella has outer membrane. You remember? The red one is outer membrane. This one is cell membrane. So then gram negative process 4 different discs. 2 discs stay on plasma membrane and 2 discs stay on outer membrane. Apart from the disc structure, they have an ankle protein called hook. Hook is a structure of flagella. It's make ankle, okay? And this disc can move, can rotate around itself, okay? When flagella move around itself 1 round, they spend 1 ATP. If they use a lot of ATP, the motor will turn around, but just like a boat motor. And the length of flagellin tail will swirl like a whip. And this make bacteria move in the opposite direction. Okay. The way bacteria synthesize flagella, they start from a disc first, and then in the disc, inside the pipe structure, they're not, they look like a tunnel. So substance inside the cytoplasm can move across tunnel to flagella, okay? This one is the secretion system that bacteria want to secrete something. But if the molecule is quite big, they probably choose the way to send the molecule past this tunnel and pass out from flagella, okay? So when the flagella grow up, the new one will go to the edge. The flagellin protein will move by this tunnel, go to the edge, and then polymerize in this area. The new flagellin will move like this. So in this area, this is all flagellin. The new flagellin is present in this area, okay? Unlike eukaryotic flagella, the new one present at the base, and then they push the old one out. This is opposite way, okay? For spatial flagella, this happen only in spirochete. Flagella is not project out of the outer membrane, but flagella stay in polyplasmic space. So this is cell membrane, and this is polyplasmic space, and the dot here is flagella. This is cross section of spirochete. Okay, flagella in spirochete has 2 functions. One is moving, let the cell movement. And the second one is flagella determine cell shape. They wrap the cells, make the cell twist like spirochete shape. Okay. Pilline or Fimbria. Well, this is confusing term. Some textbooks say Pilline and Fimbria are the same thing. Some textbooks, they classify Pilline, Fimbria are different things. I'll say like this. The Fimbria is smaller. Both Pilline and Fimbria are smaller than flagella, okay? Fimbria shorter, and when they happen on cells, they produce a lot. Function of Fimbria is used for attach to the host cells, okay? Bacteria need to attach host cells and then just begin infection, okay? If they produce the tunnel protein and the diameter of the protein, the tunnel is quite big, they probably have chance to transfer some genetic material across a tunnel. That's tunnel called pili. Okay, particularly sex pili, right? That's pili and fimbria. A bit outer from peptidoglycan membrane, we call glycocalyx. The glycocalyx is outermost structure of the prokaryotic cells. They can be capsule or slime layer. So again, some textbook they say capsule and slime layer are the same thing, but capsule here contain a pack of carbohydrate polymer packed in capsule. There are few protein inside that. Once they feel it's so tight, capsule is mean it's not good. Okay, capsule will thick and cover around the cells. Function of capsule for protect the phagocytosis of the white blood cell, right? But if the glycocalyx is loosely, not tightly packed like capsule, we call slime layer. The slimy thing in here is carbohydrate and protein, okay? When they loosely, some part of that they lose out of the cells. And this is the beginning of, well, the function of slime layer is kind of primary precursor of the biofilm production, because they can lose the slimy out and let the area cover with the film. And then within the film, there are so many bacteria living inside that, form a new colony of new community of bacteria. Community is mean like multi-species of bacteria. They live together. Okay. Bacteria with capsule, normally you see the smooth colony, and then when you stain, you probably see some empty area around the cells that capsule by negative staining. And this is biofilm formation. So bacteria starts produce glycocalyx that is slimy, and then slimy lose out, and accumulation of the waste of the slimy, then they form biofilm. Okay. Then the internal structure, then we move inside the bacterial cells. That is easy. Chromosome or nuclei. Bacteria have different from the other eukaryote. Okay. I will not say this is the truth, but most of them, not all of them, most of the bacteria has single circular chromosome. But some bacteria we know have that two circular chromosomes. Some bacteria has 11, 22, 23 chromosome. That's exceptional, but most of them are close circular chromosome. Sometimes we can find the close circular DNA, but much smaller than chromosome. We call plasmid. Okay, plasmid is the extra chromosomal DNA. All right. Well, plasmid normally carry gene that potentially benefit to bacteria. For example, toxin gene, the virulent factor, or some gene that help bacteria metabolize some toxic substance. Okay, that's the nuclear region, nuclei. For ribosome, prokaryotic ribosome are different from eukaryote, of course. Size is smaller. Ribosome of prokaryote contain 50S as the large subunit and 30S is small subunit. Okay. Small subunit comprise the rRNA 16S rRNA. S is Svedberg unit. Okay, the unit determine buoyancy of molecule in cesium chloride. Okay, large subunit comprise two different rRNAs, 5S and 23S. Okay, two of these large 50S and small 30S together we form 70S. Okay, this is bacteria, right? Next one, this is rare but can be found in some bacteria. Is granule. Bacteria has granule inside that. Some granule contain carbohydrate. Some granule contain sulfur. Some granule contain magnetic. For example, the magnetosome of bacteria in magnetobacteria. Because these bacteria need iron for growth and they live in the iron rare. So they generate iron inside the bacteria to trap the other heavy iron. Okay. Last one, endospore. Endospore is a structure of bacteria. You cannot find endospore in eukaryote. In bacteria, endospore is not for reproduction, okay? But the name spore also, spore means seed, but not for reproduction, but spore, endospore generate for let bacteria tolerate to the extreme environment or environment that harm to bacterial life. The reason they're not reproductive because one cell produce one spore. They cannot multiply, okay? But they are so rare of bacteria that can produce endospore. In fact, there are 3 genera of bacteria that can generate endospore. One is Bacillus. The second one is Clostridium. 2 of these are human pathogen. The last one is Sporosarcina. Sporosarcina is non-pathogen, okay? But in some type of bacteria, for example, in Clostridium, location of spore, well, Clostridium is bacilli, long bacilli. Position of spore can help scientists classify or identify group or type. For example, Clostridium tetani, spore present at the tip of the cells. We call like a drumstick, mighty glong, a drumstick like this. Sometimes spore present in subterminal, move from the terminal a little bit inside of the cells. Sometimes spore can be central, present in the middle of the cells, something like this. So location of spore can help them determine a big group of bacteria, okay? That's all from lecture 9. Anyone want to ask question? Are you already?
Ajahn?
Yeah.
On slide 23, you mentioned that I don't know if it was a cell or a component, but you called it a particular name.
The goat cell. Goat is mean like G-O. Cells. It is so difficult. If I zoom like this, you see the cell?
Yes.
Well, this is very difficult to see, right?
It's called God cell เหรอคะอาจารย์
ใช่เซลล์ผี
Okay. Thank you.
Ajahn, can I
not
ask
common. This is not common in real life. No one use gram stain for mycobacterium, but I made this in the lab because I want to show like that's the reason you cannot use any general stain to observe mycobacterium.
So this ghost cell is the microbacterium, right?
Yes, but the other thing like this are, well, general gram-negative bacilli.
Okay, thank you.
Dan, can I ask something on slides that you were talking about, the slide 22 อะค่ะ like I don't get when you pouring acid alcohol and then what kind of bacteria that
Okay.
decolorize.
So you need to know the slide that we need to do acid fast staining, not just from culture. We take patients sputum from a sputum from tuberculosis patient or someone that doctors suspect that it should be tuberculosis. Then they put sputum and you see the sputum on slides. On the sputum, not just only mycobacterium, tuberculosis cell, there are so many normal flora cells in your throat, okay? The circle determine the normal flora. For example, Staphylococcus, Streptococcus. You don't want to see something like this. If you want to observe the red one, then you add carbofucin and add 1 drop of orgitol. Carbofucin will move inside the cells easily. Well, carbofucin move inside all cells, all right? And when you put acid is mean hydrochloric acid. Watch the dye. Every normal bacteria will allow the watch the dye, except mycobacterium. They can keep the dye in cells. That means they tolerate acid. Okay. Once you watch,
so
yes.
Oh, so you only wash the dye from the normal cells, and then the tuberculosis bacteria will stay red because of the dye.
Exactly. That's why they call acid fast, because it tolerates acid.
Oh.
And finally, you just use methylene blue as a counter stain to make it the other bacteria has shown in different colors. You probably see some bacteria like a small dot here, maybe it's so clear, something like that. But the thing you want to observe is the red one. In fact, it's so small, but I zoom it. So this is the drawback of learning by no lab. All students, when you open at last book, they think like, oh, this is so beautiful, but in real life, it's not easy to see like this. Okay.
Okay. Thank you, ค่ะอาจารย์
ครับ